Hello, everyone. My name is Kim Hummer. I'm the research leader and curator at the National Clonal Germplasm Repository in Corvallis. I'd like to welcome you here today to tell you about our conservation efforts of temperate fruit, nut, and specialty crops. This is the third in a series of discussions of clonal germplasm repository maintenance in the National Plant Germplasm System. To take you on a little historical round, the U.S. began its initial plant introduction stations through the U.S. Bureau of Plant Industry in the late 1800s. It started introduction stations at Chico, California, Miami, Florida, and Beltsville, Maryland. People realized that gene banks to preserve and conserve this foreign material, this foreign plant material, needed to be established. So right after World War II, regional plant introduction stations were established at Pullman, Washington, Geneva, New York, Miami, Florida, Griffin, Georgia, and at Ames, Iowa. And a backup gene bank for the seed storage laboratory was established at Fort Collins, Colorado. In the 1980s, gene banks for preservation of clonal repositories were established. Our gene bank at Corvallis was one of the first established in the 1980s. The Davis Repository, Riverside, College Station or Brownwood, Texas, Miami and Mayagüez, Puerto Rico, Hilo, Hawaii, and again, Geneva, New York, were established for horticultural crops. And the National Arboretum at Washington, D.C. was established for ornamental crops. The National Clonal Germplasm Repository at Corvallis has assignment for 8 major crops: hazelnuts, strawberries, hops, mint, pears, currants and gooseberries, raspberries and blackberries, blueberries, and cranberries. In addition, we have crop wild relatives of plant materials that are either cross-compatible or graft-compatible with these 8 major genera. We also have some unusual fruit species, such as Asimina triloba, the pawpaw, which is the largest fruited plant in the North American continent, along with blue honeysuckle and some other unusual fruits, such as medlar, which is a pear and quince relative. Our crops support about a little more than $6 billion annual farm gate value for farmers. So it's a significant amount of conservation effort in our crops. Of course, we are having a pandemic right now, so our staff is either meeting in a socially distanced setting in the outside. I am here in this corner, and our other staff members, I'll introduce you in a few minutes here. My colleague, Dr. Nahla Basil, is our plant geneticist. She is responsible for our molecular laboratory. Our curator for tree and fruit nut crops is vacant. That position is vacant right now. We're in the process of recruiting for a replacement. Hopefully in January, we'll be able to do that recruitment. Jill Buchakra is our small fruit crop manager. Barb Gilmore is our field collections manager. Missy Fix is our plant distribution manager. Ryan King, pictured here with his mask, is a technical assistant for our field, and he was recently hired this late summer. And April Nyberg is the technician for Nahla Basil's laboratory. Jim Oliphant is our greenhouse manager, and he is responsible for the vaccinium and Fragaria crops. Gabriel Flores started in as a Haku intern just in September, so he's only been here when we've had pandemic conditions. And we also have several seasonal helpers, such as Laura Allen and Jamie Green. As you realize, the objectives of us in the National Plant Germplasm System is to collect, maintain, distribute, and evaluate our assigned genetic resources. I'd like to start with some of the plant exploration that I've been a party to. In our case, our gene bank was started without any pre-organized collections, so we had to obtain crop wild relatives to represent the diversity of the assigned materials. So over the years that I've been here at the repository, I've had the opportunity and been blessed to take 19 USDA-sponsored plant exploration expeditions. There's not enough time to talk about each of them, but I will talk about these highlighted ones in the next few minutes. In 2001, I was able to travel to the Russian Far East in Khabarovsk and Primory territories, starting at Vladivostok with our colleagues at the VIR station there, the Vavilov Research Institute. In 2004, we collected in Hokkaido, one of the northern islands of Japan. In 2015, several of us went to collect in 4 different national parks in Vietnam. And also, throughout the years, I've been collecting from around Oregon, right here in our backyard. And then in 2018, we went to West Virginia and Virginia to collect blueberry crop wild relatives primarily from the mountainous regions there. Also in 2006, I collected plant material from Florida of wild blueberry crop wild relatives. The Russian Far East expedition was quite interesting. Our means of travel was by this troop transport vehicle. And my colleague, Nick Borza, who is at the Philip E. Marucci Cranberry Research Station in New Jersey, who is he's a professor at Rutgers University, accompanied me to collect cranberries and blueberry relatives, along with other berry crops on this expedition. Our Russian counterparts, Andrei Sabatov, was our main botanist. His colleague, Pavel Cherebukin, came with us, and his wife, Irina. And Vera Fintova was assigned from the Moscow part of VIR to accompany our trip. And this Vera in blue was the station director for the Vavilov Research Institute, where we started our expedition. So some of the plants that we collected, Vaccinium uliginosum is a wild blueberry relative that is native in bogs. Ribes passiflorum is a black currant species relative. And we were very interested to collect black currant relatives because white pine blister rust, Crenartium rubicola, the disease that is now encroaching in the US, or has been for the past 100 years, is originally from this region. So we were hoping to find disease-resistant types. We were collecting cranberries from many bogs in both of the territories, including Vaccinium oxycoccus, which is a diploid, and a hexaploid called Vaccinium hagerupii. Jumping over to Hokkaido in 2004, our travel vehicle was a little different. We were going around Hokkaido in a Nissan SUV. It had navigation, quite different than the troop transport vehicle we went in during the Russia expedition just 3 years previously. Our colleagues from Japan, some were stationed at the Tsukuba Science City in the forestry department, and others were at Akita University and botanical experts native to Hokkaido also. My colleague Tom Davis accompanied me on this trip, and he and I represented the US. We were specifically looking for Fragaria inume, which is a significant contributor to the octoploid genome. The octoploid strawberries are the wild crop progenitors for the hybrid strawberry that we eat. This particular strawberry is a diploid. In 2007, I was able to go collecting with Paul Lyrene, who is the breeder from the University of Florida. And we were particularly looking to acquire fruit to extract seeds and cuttings of Vaccinium darrowii, one of the low-chill blueberries. And this particular species has been a significant contributor to low-chilling genes in blueberries. Breeders have been making crosses between darrowii, elliottii, another southern adapted species, and the highbush blueberry, so that they could obtain southern highbush blueberries that require less chilling in the winter and can produce flowers and fruit without having to go through a cold winter condition. In 2015, Jim Oliphant, our greenhouse manager, and I went collecting in 4 national parks in northern Vietnam. And our colleagues in Vietnam included several people from the Plant Resources Center at Hanoi. And then in each of the parks, some of their park managers accompanied us on our trip too. We were collecting latitudes and longitudes and making voucher specimens of the crop wild relatives of rubus and vaccinium. In 2018, I, accompanied by Jill Buchakra of our unit, went with Jim Ballington, who is from North Carolina. He's a retired breeder, a small fruits breeder. And Paul Lyrene is the breeder from the University of Florida, who was working on the low-chill blueberries. We are standing on Spruce Mountain, the highest point in West Virginia. We were also looking at the Dolly Sods research area, and we were collecting native blueberries from these regions. Vaccinium erythrocarpum is a red-fruited type of blueberry. It's really dark red fruit, and you can see the beautiful foliage it has in a fall condition. I've also collected North American Vaccinium from Oregon and the Intermountain West. There's a section Myrtillus that includes the European Vaccinium myrtillus, but there is a disjunct species of Myrtillus in Colorado and in Oregon. And besides that, there's Vaccinium ovalifolium, Vaccinium membranaceum, Vaccinium deliciosum, and Scoparium, and the red-fruited Vaccinium parvifolium. We've also collected the Oh Hello Berry from Hawaii, Vaccinium reticulatum. That is the low-growing form, and the tall-growing form, Vaccinium collicinum, is also there, both on the island of Hawaii and the island of Maui. We worked with our colleague Francis Z in developing 3 different cultivars of Ohalo berry. The next topic I'd like to mention is the maintenance and distribution aspects of our gene bank in Corvallis. We have between 12,000 and 13,000 accessions. It's been cumulative over the years, and we distribute between 5,000 and 10,000 accessions depending on the year. Our berry crops are kept in screen houses. The screen prevents aphids from entering. It's an aphid exclusion screen. Inside our screen houses, this one is House 5, are raspberries and blackberries. Jill Buchakra manages these crops, and it's a challenge. They have a lot of prickers, like we were talking the other week about prickers in pineapple. Blackberries and raspberries is another crop where you need appropriate PPE, gloves and long sleeves. And we have a saying at our repository saying, no blood for rubus. We don't want to get prickles in our hands. We also have cumulus, the hop germplasm, and strawberries. We have the strawberry collection includes 2.5 screen houses worth now. We have about 2,000, more than 2,000 accessions of strawberries, including heritage strawberry cultivars such as Marshall. Marshall is still frequently requested because it's no longer very available through commercial means, but people really like the flavor of this fruit. And in fact, Marshall was one of the founding clones of the UC Davis strawberry program initially, and also the Driscoll's program. Driscoll's, of course, being now quite an international berry company. The challenge with Marshall is that it doesn't pass the drip test. If you put 2 Marshall fruit that are ripe on top of each other, the juice is going to squish out, which is great if you're eating strawberries straight from the field. But if you want to transport these flavorful fruit to market, it presents a challenge. So people use this for a basis to obtain flavor and have tried to get more firmness in the berry, disease resistance, and all other good quality traits. Hazelnuts is another one of our main crops. The European hazel, and this happens to have red bracts. It has bracts that open, and the nuts are large, and they tend to dehisce in the fall. This particular species is Corylus cornuta. It's a Native American species, and you see it has clasping bracts. Hazelnuts are an unusual plant because they bloom in the middle of winter. The female flowers are right at the base of the male catkins. So this is a monoecious type of a flower where the female flowers are separate from the male. And the female is just this little red, very inconspicuous flower right at the top. And I talked to Chris Walters about preserving pollen because this is, we can take catkins and force the catkins and extract the pollen fairly readily from hazelnuts. So it would be a good way to back up half the germplasm, anyway, at Fort Collins, Colorado. Our pear collection includes more than 2,000 different accessions. You can see it's not like having a Bartlett orchard where it's one type of a cultivar. We have many different fruits and they ripen at different times during the year, from the Chinese pea pear to Asian cultivars. We have Conference, which is a long-necked European type fruit. We have a Yubaline, which is a very large fruited pear, and everything in between from Asia to Europe. Taiwan researchers recently called us because their forests are being taken down and housing developments are being put up. They asked us if they could repatriate Pyrus Kawakamii, which is the evergreen pear. This pear was one that Mel Westwood collected in Taiwan back in 1981, and we sent them seed and cuttings of this species and tissue culture plants that they could repatriate to Taiwan. Here's another interesting story. Pears are grown on rootstocks, and researchers have been looking for dwarfing rootstocks over the years. One of the heritage dwarfing rootstocks is the little dwarf, the little green dwarf, Le Nain Vert. This Le Nain Vert tree was planted in 1984, so it's 3 years old. And the little boy here is 6 years old in this photograph. 10 years later, the boy is now 16 years old, and the tree is still quite dwarf at 13 years. Now it's 30 years later, and 27 years for Le Nain Vert, and it still has a much shorter stature than other trees in the background. There's a lot of work that goes on in the dormant season and into spring with 2,000 pear trees, 76 different rows, and the rows are 50 plants long. They're very big. We have used the kind of cutting devices that John Priest demonstrated in the Davis discussion, but a lot of our pruning is done by pneumatic pruners or chainsaws to bring down the height of our collection so they can be manageable. Also in the dormant season, we need to prune back blueberries and other woody tree plants in our collection. Joseph Postman, our curator, has recently retired, as I mentioned, but he has left us a few gems to find as we go forward. I mentioned about the blue honeysuckle. This is sort of like a blueberry, but it's a little more sour. It ripens earlier than blueberries, and it's one, if there could be harvesters, some of the farmers are interested in, that produce blueberries, are interested perhaps in using the equipment that they have for harvesting, if they can use it for another crop, such as blue honeysuckle. They're interested to diversify their fruit production. Maxine Thompson, one of the emeritus professors here and one of our founding mothers of our gene bank, is a breeder for blue honeysuckle and is selecting types that are adapted to the Corvallis region. This particular type I have here, Marina, was selected by Maria Plankovna at St. Petersburg. So it is a Russian type. It is selected from material in the Kuril Islands and in Kamchatka. So it's a very cold hardy type. Maxine was looking to breed more of the Japanese type, which is more torpedo shaped than the long cylindrical Russian type. This picture shows you some of the diversity of ribes, the currants and gooseberries in our collection. They have beautiful flowers. The pink flowering currant is one of the earliest spring flowers. There are white fruited forms with long pendulous racemes. Ribes viburnifolium, the evergreen currant, is native to the Catalina Islands, just off of California. And some of these, like Ribes menziesii, is native in the Siskiyou Mountains. These gooseberries, Ribes menziesii, Ribes pinetorum, produce gooseberries that have sort of a Sputnik-like shape with prickers coming out. And actually, these fruits were used by native peoples who peeled off the spikes and ate the inside. I mentioned about ohelo berry and the selection of red button, kilauea, and nene are three different cultivars that Francis C. selected and we're maintaining in our gene bank here. This project was done as one of the early SCRI grants, one of the first ones that was given out. Many of the seeds of our genera are long-lived. We keep seeds of the species of our collections in minus 18 degree freezers, and we're working with NLGRP at Fort Collins to back up these seed collections as well. This is an example of rubus seeds, and you can see Rubus megalococcus, which is the largest seeded rubus in our collection. Rubus ellipticus came from China. It's the smallest one that we could find. They do have very reticulate seed coats, and they're very interesting seeds to look at. This particular megalococcus is 80 times the size of the Rubus ellipticus seed. We have sent our seeds back up not only to NLGRP in Fort Collins, but excess seed has been sent up to Svalbard at the Svalbard Seed Vault in Norway. As Robert Kruger mentioned in his discussion of citrus, there are a lot of germplasm threats. One of the more recent threats is eastern filbert blight. During the past 15 years, this fungus has now established itself up and down the Willamette Valley here in Oregon. And we now have to manage our collections for this fungus. We have to spray in the early spring, and we cut out diseased branches. We have established the core collection down at Parlier, California. We greatly appreciate the collaboration with Dr. John Priest and Claire Heinz in the management of our core germplasm collection down at Parlier. And it's very important to have that there, so we have a second location backup for these important cultivars. In hops, we've had powdery mildew fungus appear in the last several decades, and hop stunt viroid. So the hops that we maintain are in this greenhouse. For pears, the temperature has changed, so it's been a warmer spring, and when the pears are blooming, they are now subjected to fire blight. This never used to be a problem 20 years ago. It just wasn't that warm, and the fire blight organism didn't bother our trees. Other diseases and pests that we've had are the red scarlet mite, pear blister canker, pear decline, which is a phytoplasma, and Xylella leaf scorch. We have recently found black currant reversion virus in our collection, but we do not have the big bud mite, eriophyid mite, that is the vector of this virus. White pine blister rust continues to be a problem for currants and gooseberries across the United States. Our plant pathology colleagues and virologists have been busy discovering new viruses in blueberries. We do have blueberry shock virus in our field collection, and we are evaluating for plants that are slow to get shock virus. We do not have scorch virus here. That is native to back east. And once again, we keep our blueberry collection under screen away from viruses. The screenhouse blueberries have tested negative for viruses and other diseases. We distribute from the screenhouse only. Some of the studies are looking at blueberry fruit, and so where people are just collecting fruit and sampling it for evaluation purposes, that can be shipped. Phytophthora ramorum, sudden oak death, can be carried in blueberry plants, so we do need to be inspected annually, and thus far we have not had sudden oak death in our collections in Corvallis. We perform pathogen testing through visual inspection, biological assay, ELISA every 3 to 5 years, and PCR as new techniques are determined for pathogen testing. So in evaluation, we have a very active molecular marker laboratory. Dr. Nahla Basil is in charge of this laboratory. They have obtained fingerprints for helping us identify our plant collections. Nahla was one of the first people to get molecular markers into BRIN, and our blueberry fingerprints for this early study were able to separate the northern high bush, the wild types, and remember I was talking about combining the wild, making crosses of the wild with the northern types to get the southern high bush. And we can readily distinguish these groups using the fingerprinting studies with simple sequence repeat markers or microsatellite markers, as they're called. And we do a lot of cladograms and phenologies, and we're used to looking at this two-dimensional type of separation. But we had a graduate student, Katie Carter, who looked at a target capture sequencing probe design in rubus. She wanted to estimate the phylogenetic relationships in rubus. So we realized that we could develop probes for 926 black raspberry single copy nuclear loci. And she also looked up 257 low copy nuclear loci conserved across Rosaceae, including from apples, peaches, and the strawberry genomes. And with more than 1,000 genes, she began looking at the relationships of 94 different rubus species. Rubus is highly complex, and so she developed a network. And it's multidimensional. If we could just see beyond 3 dimensions here, we'll have to use our imagination. The outgroups are in group 1. Raspberries are over here in group 7 for the majority. And blackberries are here in this blue region. If you look closely, loganberry, which is a known cross of half raspberry, red raspberry, and blackberry pollen, it appears right in the middle between the blackberries and raspberries. Cultivars such as Marion and Boysen are much more related to the blackberries of their pedigree, they have a tiny bit of raspberry in their second generation out, grandfathers and great-grandfathers and great-grandmothers. So they're mostly blackberry type genetics. So it's really interesting to see the complex structure and the reticulation involved in the evolution of rubus species. We're still chewing over this and we're learning a lot, and we're going to be looking more specifically about the edges of these interactions. I wanted to leave you with one other complex genome, and that has to do with the strawberries. Recently, Steve Knapp and his group and his graduate student Picot et al. Looked at the social network analysis of the genealogy of strawberry. They retraced the wild roots of heirloom and modern cultivars. Strawberry is interesting because it's only several hundred years old. It started back in the mid-1700s after a species from Chile, the Chilean beach strawberry, was taken to a garden at Versailles in France. And in France, there was already the Virginian strawberry, Fragaria virginiana. So this very smart botanist, who was very young, 19 years old, Antoine Duchesne, recognized that a hybrid strawberry that had much larger leaves and much larger fruit developed from the mother plant of a chiloensis with pollen from the virginiana. And that produced the first cross of hybrid strawberry, and that's the one we eat. So since then, breeders have been making crosses, and Picot et al., under Steve Knapp's guidance, developed this social network of the strawberries, looking back at the heritage and heirloom strawberries and the relationships of these plants. We do have Madame Moutot, which is one of the oldest strawberries in existence, strawberry cultivars. And it's very interesting to see the relationship here. This is much different than the cladograms we're used to working with. So we're looking into the future here by examining the past very closely. Well, we would like to welcome you to come to Corvallis and visit our gene bank and work with our staff members. But at the moment, we are under a pandemic situation. Once the vaccine is out and the pandemic is over, we would be glad to have you guys come and visit. We have in common with you our mission of preserving plant genetic resources for all people for all time. So thanks very much for listening, and we'll talk to you very soon.